Samik, I'm really excited to get to sit down with you today. Thank you for taking the time. You are the Chief Scientific Officer of Cabaletta Bio, a company that's potentially going to bring curative CAR T-cell therapies into autoimmune diseases. Oversee all of the discovery, preclinical, computational biology, and translational medicine. What exactly does that mean? Because a lot of people wonder, like, what does a Chief Scientific Officer actually do?
Yeah, so the different terms, discovery, preclinical, translational medicine, computational biology, three of them refer to different phases of drug development. The discovery and preclinical are kind of the concept of a drug, everything from what type of drug do you want to develop, how do you want to target. In our case, we want to target B cells. And after you have a few candidates, that's kind of the discovery phase. You have one that looks, let's say, most promising. You take that to the preclinical de-risking, essentially. Basically, that your drug is as safe as it can be before you put it into clinical or patient testing. And that involves, talking to the FDA, having an investigational drug application, they clear it, and then you can go into patients. And the translational medicine side is kind of the assays that we do on patient samples to explain, does this drug work? Does it not work? If so, why? And also to kind of work up any kind of adverse events should they happen. A lot of the same assays you do preclinically, but on patient samples from your clinical trials. And computational biology is kind of a thread throughout the entire process, and that really has to do with kind of high throughput data. So think RNA sequencing, DNA sequencing, whole exome sequencing, that kind of thing. We also do a lot of serum analyses with, I'd call it mid-throughput protein analysis, and the computational biology team helps us make sense of all that.
Cool. So like from your perspective as CSO, what role do you play in all of that?
Yeah, so on the discovery side, some of it's coming up with an idea as to what should be our next drug. We haven't publicly disclosed anything just yet, so I can't really share that. And then working with our teams to say, is this a good idea? Is this not a good idea? What would the best candidate be for this type of drug? Overseeing the experiments, or overseeing the teams that oversee those experiments. We have a vice president of preclinical research who kind of does a lot of that with some of my guidance. On the translational medicine side, so we have a chief medical officer who oversees all of our clinical trials. And for a lot of clinical trials, you have things like primary endpoints, secondary endpoints, exploratory endpoints. The primary endpoints from most phase 1 trials are like, is this drug safe? They're a bunch more than that. Secondary endpoints are like, does it work? Exploratory could be like, how does it work? And so a lot of what we do on the translational medicine side is kind of in the exploratory bucket. How does this drug work? So my job is to help. Again, we have a head of translational medicine as well, who I work very closely with, and kind of design, what are the things that we want to ask, and what are these samples that we want to take from people after we infuse them with Rese-cel or a CAR T, the assays that we want to run, and how we want to put that together. Now,
did you always believe that you would get to this point in your career?
Not initially, no.
What did you imagine for yourself?
I always figured I'd have a job like doing some kind of, overseeing a lab or something within a company. I saw that model at Merck where you scientist, director, whatever the term is, but overseeing, let's say, a smallish group or small to mid-sized group kind of doing research. At the time, I thought it'd be more discovery that would, help generate new ideas, products, whatever. That was kind of what I envisioned initially. So it's kind of, very happy I'm CSO, but it wasn't initially what I anticipated.
Okay. So you went from a medical resident to CSO we were kind of calculating it, like 12-ish to 15 years, somewhere in that range. Yeah. And that can typically take 20 to 30 years for some people. So what do you think actually contributed to that accelerated pathway for you?
That's a good question. I think it's a few things. Some of it's luck. So after medical school, I ended up doing human immunology research. So I was a postdoc, essentially, for about 4 years at Penn. Now I ended up in a great place. I was with Jim Riley at the University of Pennsylvania under Paul June's group. And so, I got to kind of see all the CAR T stuff happen as it unfolded, kind of in real time.
Like a great place to learn, right?
Yeah. And I was there from 2005 to 2009. And it's a great place to learn immunology. I got lucky on all the projects I worked on. I got a publication relatively quickly. I also got very good guidance. I had to do residency at some point, and the guidance I got was to do a clinical pathology residency. So I think when most people think pathology, there are two types of pathology, anatomic and clinical. And most people think by anatomic, which is like, hey, what is this autopsy? Why did this person die? Or we did a biopsy. Does this person have cancer? And that is 80% of probably most pathology practice in the United States. Clinical pathology, as I was informed, was called the Physician Scientist Refuge. So it was a place, it's a lot of like the same lab techniques you learn as like a postdoc or scientist, like flow, Western blots, all that kind of stuff, but applied to clinical medicine. And so the folks who gave me this guidance said that, you kind of know how to be a scientist. You're a scientist already. Residency will be a little bit easier for you. Also, if you want to do research, it's a great way to do research because the clinical stuff will be a little bit easier to get because you have a scientific background. So, that was pretty good advice. So I ended up doing that for a few years, 3 years. The residence itself is shorter. So a normal anatomic and clinical path residency is about 4 years. But if you only do one of them, it's 3 years. So you save time there as well. And you can spend more time in the lab. So I got lucky with where I ended up for my postdoc, and I got really lucky with my residency because I could kind of still focus on research. Then, level luck here. I got a very unique kind of transitional position at the NIH. So most positions at the NIH are funded by the NIH because that's how that kind of works. My interest was in Treg research. Actually, I'm still interested in Treg research, but I had this opportunity to work with a company, or actually for a company technically. So
This was while you were at the NIH?
While I was at the NIH.
Okay.
So I was in Ethan Shevak's group in the laboratory of immunology at the NIH. Ethan had worked out what's called a cooperative research and development agreement with Boehringer Ingelheim. And so basically they would pay us, I forget the exact amount, but something, let's say in the R21 range, I could have a grant, like $250,000 a year, give or take. And our goal was to develop Treg biologics. So think antibodies that can modulate Treg function, essentially. Either up or down for cancer, up for autoimmune disease, essentially. But it'd be entirely independent of anything in the NIH, which is kind of unusual.
That's unique.
Yeah, it was really
unusual.
unusual. And so, I would be paid under that. I could hire a technician, great.
Okay.
And I could do half of whatever else I wanted to do while developing this kind of screening platform or discovery platform for Boehringer Ingelheim. So that was really my first kind of real foray into drug development from very early phase. So I learned a lot about high throughput screening, what's useful for, let's say, the initial phase for looking at candidates and subsequent confirmatory stages. So it was something I hadn't really been exposed to before. I didn't really know anything about antibody discovery. And I got really exposed to that very quickly. The things that, if you're in discovery function in a large company, what do you look for? How do you do it? So that was very, very instructive.
That is so unique, because you were working at the NIH, but you were actually working for a drug company, essentially, as an extension of them. Basically, we're doing what you're doing now, drug development early on and finding new targets and things like that. But there was also something that you had mentioned, or that you've talked about in the past, in your postdoc, I think it was, that was also really unique. I'd love for you to share a little bit of the advice that you've gotten at that point. I've talked to a lot of people who are doing their PhD or are in a postdoc and, they're trying to figure out how to transition into industry and maybe are not getting or haven't gotten guidance on really what it takes to move into industry. So that part about the NIH is really unique, but I think you also had a little bit of guidance earlier on too that helped along the way as well.
So the lab that I was working in at the time, there were lots of junior faculty. So think people who just gotten their first assistant professor positions, mostly physicians, not exclusively physicians. And, they're trying to get their labs established and become independent on their own. So these are guys, or guys and girls who are let's say 5 to 7 years ahead.
When you say ahead of you mean in their post, they're in their careers.
And, we were all still working in the lab, which was really nice. Because I'd come back and talk to my boss, and he'd give me this laundry list of experiments. We have a question on how does this Treg work or something like that. How do we check if it works? And there'd be 15 different experiments, and I'd sit there looking at them, and these other folks would be why are you looking at these 10 experiments? I gotta do these to no, you don't have to do that. First of all, get rid of half of this. You don't need to do any of that. You need to do these 3 things. And it was kind of an iterative process. I'd come back with lots of experiments, they trim them down, and they'd say, you have to do these things to get your paper published. And they taught me really to think in papers. And my boss did as well. That was very helpful. Kind of more on the kind of actionable, what you need to do, A, B, C, D. They were also very big on encouraging that, you don't need to, I think there's a phrase I've heard. A lot of PIs have CNS disease. So it's cell nature science as to where to publish. And what I was told was you need to get 3 to 5 figures to get your first author and get out.
Okay.
And that's what that is.
Okay.
And they were very, very prescriptive, which I benefited from immensely.
Yeah.
So here's what you need to do.
Okay.
Here's how you assemble paper. You can tell it in 4 figures. If you can tell it in 3, even better. And that was excellent advice on how to get out and how to move and how to make sense.
Yeah.
Very
so
hands-on,
smart.
very hands-on.
Yeah, that's so smart. So they basically were giving you advice on like how to get published, essentially,
Yeah.
but not just how to get published, but like how to get the answers to the questions that you had on the table a lot faster than you probably would have had you done all 10, 15 experiments.
No. I take that to me. I take that with me to this day because it's like, you can always do more. You can always do extra. In some situations, it's warranted, but it's really not. It's really like, here are the 3 or 4 things you need to do. You don't need to figure 10x. That's not important. You need to figures 1, 2, 3, 4. You need the most relevant questions answered first.
Yeah.
That was very helpful. It was very specific guidance, and they were very prescriptive. And I benefited greatly from like, do this, not that. Do these things as fast as you can, reliably and reproducibly, and all that kind of stuff. So it was very directed.
Because I imagine that's what you brought into that NIH role, and then it's really probably what you bring into your current role today.
I don't like extra, so I think it helps focus you a little bit. So I've been again fortunate, and sometimes like when we design experiments, we'll talk about like what are we trying to answer and what's the best way to answer it, and there's like lots of things we can do. And then you start adding all the conditions and controls, and you're like, wow. And then you think about it, and I think about the person doing it, and I'm like, that experiment sucks. There's no way I can get someone to do that. That's really bad. I'm being really mean. So like, how do we cut this down, or how do we break it apart so it's more manageable?
I'd love to transition over to your time at Merck, because you and I both lived through the immuno-oncology phase, me at Bristol Myers Squibb, you at Merck. So tell us a little bit about your experience there, what that was like. I remember it was just like T for T. Everybody was just trying to get to the market first for every different indication. And now I think pembrolizumab under Keytruda has 19 indications under it now, something like that. So tell us a little bit about that time and how that really shaped you.
That was great. So I happened to work at Merck at their Boston site. I started in 2014, and Pembro hadn't gotten its first approval yet. I think the path was pretty much there. And I was in the discovery function as a physician, which is a little unusual. And my job essentially was to take late bio assets or standard of cares and see what would make sense to rationally combine with the PD-1 inhibitor. I had a small team, like 5-ish, 7-ish people. And the idea was to do a bunch of kind of preclinical experiments to kind of inform what the phase 2 trials would be. So an idea would be, we want to combine a standard of care that is alkylating therapy with a PD-1 inhibitor. How do you sequence them? Do you do them simultaneously? Do you do one before the other? And which one do you do before, kind of that mechanics, and what's the immunological rationale, if you will. And that's what the standard of care is. And with, I assume with BMS, you basically saw a chain latex and took every single co-stimulatory molecule or negative checkpoint inhibitor and tried to make an antibody out of it, basically.
Yeah.
With those coming out of discovery, how would those combine with PD-1 blockade? It was fun. It was very scientifically rigorous. So people really wanted to know the mechanism as to why two things would synergize. Now, eventually we figured out it had to do with chemoinflammation, with the standard of care, therapy, and not all T-cell agonists would necessarily, or T-cell active agents would necessarily synergize. There were some, but not all. And also, there's only so much juice you can essentially squeeze out of a T-cell. So that was kind of interesting to learn as well. But I think more broadly, I think what surprised me was industry science can be very scientific and very mechanistic, which I didn't quite appreciate. And then it was very line of sight, it was very directed. For combining an alkylating therapy with a PD-1 inhibitor, and you don't know how to do it, the reason we're doing these, let's say, mouse experiments or whatever, in vitro experiments, is to tell the clinical team, hey, you want to give the alkylating therapy first, then you come up with PD-1, then you come up with Pembro. It's very clear questions we're trying to answer with very clear outcomes. And I think that's both kind of cool, right?
Yeah.
It's also very motivating for yourself and for the team that you manage, so that's very helpful.
So you're essentially like creating the phase 2 strategy. Most of these could go right into phase 2?
Yeah, because
like it was a phase 2 strategy. You were de-risking it preclinically.
Well, it wasn't even... So the phase 1 trials already been done, so we kind of knew the safety profile.
Yeah.
It was just how do you combine them? Yeah.
Yeah.
So
you're essentially creating like phase 2 strategy, the dosing.
Well, you're helping inform that. You're helping.
Well, to a degree.
Yeah.
Which is awesome. And then you also, so tell me a little bit about like, you've gotten to where you are because of your accessibility to a lot of decision making and some of those, like what you were just saying, like around very directed line of sight, like you could see very specific questions, outcomes that were going to come from it. So like, Talk a little bit about like what role that plays and why that's important for scientists to be able to understand, because I think that often gets, kind of pushed to the wayside. Like we focus so much on the science. We want, we obviously are all wanting to get science into patients, but like there's a lot that goes into deciding why this mechanism, why this combination, why, or just in general, like why do people invest in certain things? So I'm just kind of curious, like, can you talk a little bit about some of the decision making that you were exposed to? Yeah. That really plays a role and why that's important for people to understand.
So I think, there are a few levels. What is the point of this assay? What is the point of this experiment? Does this assay participate in somehow making a drug or getting something into a person? Or if it's in a person, to tell us why it works. So I think that's kind of a base level. I think it's very important if you're at the bench, if you're managing some of the bench, to be able to understand and then communicate very effectively. And I learned a lot of that again. I saw a lot of that in work very, very clearly firsthand. I think other levels more strategically, and if you're junior, you may not be participating in those discussions. That's fine. That's kind of how career development works. But I think the biggest thing I got from Merck was being in the room when those decisions were being made. So I reported into a guy named Michael Rosenzweig. I think at the time he was an associate vice president at Merck, kind of ran a very large remit. So immuno-oncology, small molecule oncology, and then kind of all immunology discovery. Yeah. So like 70-ish, 83, I don't exactly know the exact number, but very large group. And I probably was his most junior direct report by far. And so, but I get to be in the rooms when kind of, his team met, his leadership team met. And it was very interesting to see, if we're considering a new asset, is it safe preclinically? Is there a rationale for it? If we're considering an acquisition or partnering, what is the scientific rationale? And again, I can contribute like, hey, this works with the T cell, this doesn't work with the T cell, that kind of thing. But it's very interesting to see how everyone else weighed in. And just to be there was extremely informative.
So tell me more about that. Like what did you listen for? What were the things? What was it like to be basically like a fly on the wall?
A lot of it was, there are folks who were like 20 years ahead of me, right, in that group, who would disagree with, let's say, Michael, or they have a discussion as to, okay, here's an asset, we have a very similar asset. Here are the pros and cons. Here's how long it'll take for development. Does it really make sense? Then you occasionally get the business folks weigh in. It doesn't make a lot. And again, I can't contribute to that, especially at that point. But it was very interesting to see how the decision made, irrespective of what the decision was. Like, you want to go for it, you don't want to go for it, whatever. Or you want to de-risk it this way. That, just being around for that, I can't think of anything more instructive or informative. And again, I don't know if people teach classes on this kind of thing, but it was just, you were like, okay, this is how these decisions are made. Yeah. And again, it was very eye-opening to kind of sit there and just kind of hear it all. My boss was also very nice and gave me the opportunity to evaluate lots of assets that came through. Everyone at that time had an idea of, this will combine great with Pembro. 80% of them. Some of them were so bad.
But that happens, right? You trial everything, right? You consider everything. That's what drug discovery is.
There was one time, like, they would come from academic sites, companies. I remember once I was listening to a pitch, and I was like, and I thought to myself, how did this man get $20 million? Like, who gave them this to fund this idea?
Oh, I know a lot of scientists who have that thought. I've spoken to a lot of them, and they're like, how did this get funded?
Anyway, that's not here nor there. But it was, again, being involved in that process. My boss was pretty, not micromanaging. Like, he'd be there if I need to, like, ask him scientific stuff or, like, discuss something. But he's very just like, here's the structure, here's your remit. It's pretty broad. Come to me as you see fit, or not. Mm-hmm. And you'll be in the room.
Well, it's interesting that you say that because, we used to debate a lot. I shouldn't say we used to debate, but there was always like kind of this constant debate where I was that, should we include, should the governance meetings be kind of like open meetings? When I say governance, I mean like where those decisions get made, like when the teams are presenting about their asset, should we advance it? Should we continue to invest in it?
Sure.
Do we agree with the path that the team has laid out, given all the risks, like a lot of the drug development stuff that you talked about earlier. And we used to debate a lot, like, should we have those be more open when, some of it is a little bit more, we want to keep it more confidential and like to a closer knit group. But like, then there was the advocacy for like, people are like, how are we ever going to learn how to do this if we never are present for those conversations? Yeah. So what I'm hearing you say is like, it's actually really very important to be part of those, to understand like what are all of the factors that get taken into consideration when we're making decisions about, whatever the question is on.
Exactly.
Your question a lot of times was, what do we combine with Pembro? But there's often other questions that are on the table. So thank you for, I appreciate you sharing that, because I think it's so important for people to understand that decision making isn't easy, and there's often a lot of people that weigh in. And I think also the other thing that really struck me with what you said is your AVP, the manager that you reported to, really took into consideration everybody kind of have an opinion about things, which I think is also really important for that critical thinking skills and things like that.
No, it was again, I was very fortunate, lucky, whatever term you want to use. And he just let me hang out, and he let me say whatever I want. Could be good or bad. Most of the time I had the sense not to say too much. I kind of recognized where I was in the pecking order. But again, just to be in the room.
Yeah.
And that's how John is thinking about something, or Sasha's thinking oh, okay, I see what's going on. I see why they think that way. Yeah.
So now, as CSO, what of those values and behaviors have you really pulled forward into how you run your team, how you build your team, how you run your team now? Give us a peek behind the curtain of what it's like to work for Samik.
you should interview my direct reports just to do a comparison. I have a staff meeting every few weeks, and it's people who report to me and some of their direct reports, along with our project manager. It's a mix of everything from science, HR, decision-making, whatever. And I think the one thing I try to do is be very transparent as to what I share, as to why we're contemplating things scientifically or programmatically or even from a bigger corporate perspective. So that I try to be very open about what we're doing. I think as open as one can be with more junior folks. But I also think the decision-making process, or why does something work? Why does something not work? That should be open to everyone. Again, there has to be a certain cutoff. And I think my teams are very clear. I'm hey, these are my thoughts. I'm open to any other suggestion, thought, whatever. I'm not married to this. Or if I'm asking a question, here's a question, here's my answer. Is there a faster or better answer? Because I'm really interested in if there's a faster answer.
Yeah. Do you have any examples of that happened?
So our first patient that we treated with Rosacell, that's a case report that came out a few years ago in molecular therapy. And I still remember when we were we dosed a patient, we were pulling all the data together, a lot of the translational data. And we had a pretty big meeting to it was me and the entire translational and computational biology teams to figure out what was going on. So we're all in a room, so I forget exactly where we were, but we were in this room, 15-ish people, and some people had slides prepared based on the data they analyzed, and we were just kind of doing stuff in real time. And I think some of the more junior folks in that group got to see how we started coming up with conclusions or hypotheses around how Rosacell is working. We had some idea from how CD19 CAR T cells would work in autoimmune patients from the Schett data that was published. But mechanistically, we kind of uncovered a few things that actually came up in that meeting that we had never really thought about before, but everyone got to see, especially the junior folks, how we kind of put that together. So one thing that we reported on is typically people look at CAR T persistence, especially in oncology. CAR T persistence is everything. The more CAR T persistence you have, means drug is working, blah, 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 patients get better, all that stuff. I am vastly oversimplifying all this, but that's a belief. And what we found was in autoimmune patients, maybe the majority or a lot of them, before we see persistence, that's kind of the first read of efficacy, we see this spike in a cytokine called Interferon gamma. And that happens about a week before persistence goes up in the peripheral blood. And we think that results from the CAR T cells going into the body, engaging the B cell target, killing those B cells, getting activated, and then secreting interferon gamma. And that eventually builds up and becomes visible. We can detect it in the peripheral blood. And that happens before the cells start to divide more, and we get that persistence peak about a week later. But that was all kind of figured out in that meeting. There's some other cytokines that go up in response to interferon gamma, CXCL10, also known as IP10, and IL-12G40 as well. But that was all figured out in real time with a pretty big group.
I was just going to say, how big of a group did you have in a room? And
I want to say 15.
Were you whiteboarding this?
Yeah, we had a screen, we had whiteboard, we had people putting plots on in real time, cool. Hold on. And again, it just kind of occurred to us, all at once. So that's one example. And again, there's proof of that, and that's kind of borne out over the patients that we've dosed since, that there's this kind of mechanistic sequence. And that was all determined. It wasn't just me looking at slides and prepared. It was a collective, collaborative effort. The other things where we've talked more as a kind of a research leadership team as to why something doesn't work or what are hypotheses for, let's say, differential, we don't really have differential outcomes, but differential proliferation or cytokine profiles in different types of indications. And this is something we just kind of discuss and like working applications. What do you do with this? What does this mean? I have my thoughts, obviously, but I'm not too wedded to them. And there's certain things I am, but like for the most part, not too much. And so I'm kind of curious to what people have to say. Don't really need wrong answers.
Well, that's the thing about science is like it's all stuff we've never, we're learning as we go, right? Yeah. So there can't be fully wrong answers.
No.
Maybe there can be once they're proven.
Well, you kind of give people the freedom. They're like, all right, this is what I think is happening. Like, all right, that's cool. Like, how would we test for that? What do you think is the mechanism for that? So again, we try to have that. I try to have those meetings be very collaborative in that sense. The other, trying to think of other things that aren't science. We do a lot of HR stuff publicly in that forum. We discuss how we want to kind of standards for different levels of promotions and stuff like that.
So a lot of transparency around that.
Yeah. I think it helps.
Totally.
It also makes it easier for me. I don't have to do as much work. That's a side benefit. I don't like talk.
So what are, actually, since we're on the topic of Cabaletta, what are you most excited about for the future of Cabaletta at this point? There's a lot happening for you guys. I know you have like a potential BLA coming in 2027.
That's super exciting. Like the fact that you could have start the approval process, right? Like that's huge, right? And again, our Resi-cel seems to work, it works well, a lot of indication, but it seems to work really well in myositis, the dermatomyositis and antisynthetase subtype specifically. So that's super exciting. Outside of that, on a more, scientific basis, I think a lot of the data that we're generating without preconditioning is very interesting.
Yeah, super cool.
So again, as you know, in the oncology setting, everyone uses fludarabine and cyclophosphamide, chemotherapeutic agents. They make room, if you will, in patients with hematologic malignancies. And we wanted to explore if that was, needed in autoimmune disease, because the diseases are very different. And again, I'm lumping all autoimmune diseases together. There's lots of differences in between them, among
absolutely.
them. Oversimplifying. But we've generated some data in pemphigus vulgaris specifically, and it's been really amazing to see that you can get very similar persistence in patients without conditioning, persistence of CAR T cells compared to what you see in patients with conditioning. Very similar. If not, statistically, they aren't different. So that was just amazing. We weren't expecting that result. So that's super cool. And as we get more and more data there with more pemphigus patients, we've now opened up this non-conditioning option for patients with lupus. Excited to see the data there as well. So that's kind of really, really exciting.
That is really, I was reading about it and I was like, oh my gosh, this is like amazing, especially for women who are primarily impacted by, I shouldn't say it doesn't affect men, but as we know, women are disproportionately impacted. And so the fact that they wouldn't have to get preconditioning, all the fertility issues that come along with that, and other issues that come along with that, it could be completely game-changing for getting CAR T therapy.
No, absolutely. Actually, I'll tell you a funny story. So we presented that data, ESGCT, so the European Society of Gene and Cell Therapy, last year in October. And so it's like a late breaker talk. So I'm giving this talk. It's pretty well received. And then, afterwards, talking to folks. And there's a woman who I'm not going to name. She is a hem-onc doctor in Europe. Gives CAR T cells therapies, gives CAR T therapies to pediatric patients mostly. And she says to me, I can give CAR T to women now. It was, she was so excited about the opportunity to give CAR T therapy to, she's referring to this in this case. That was immediately where her mind went, and she was so excited about it.
It's game changing. It's going to be amazing for patients. CAR T as a whole for autoimmune disease with the whole reset of the immune system and all of that is game changing. But the fact that they don't have to get any of the chemo beforehand is amazing.
No, it's potentially really huge. And again, I think, as you mentioned, fertility, especially in women, and again, autoimmune disease affects more, let's say, younger women compared to, let's say, cancer. That's huge. That's huge.
And not only that, it's the logistics of it all too, because they get admitted for that.
Exactly.
It's a couple of days.
Well, I was telling someone, I was like, well, you now have, because we do 3 days of conditioning therapy, and then you have your infusion day. So you go from 4 visits to 1 visit. That's a lot easier. That's simpler.
Huge. Yeah, it could be awesome for patients for sure. I wanted to ask you questions about advice for people who are watching, who potentially would want to, move into industry, PhDs in particular, other scientists in particular, just to kind of round out the conversation, what do you believe is the future of scientific education?
Oh, that's a big one.
It is. Like, what do you think that's, like, just given your training and your postdoc, what should it look like? Like, should we be changing how we're doing things when it comes to PhD training and postdoc?
You know, I don't know what the average is in the United States for a PhD, for someone who gets a PhD in some kind of biomedical thing, but I want to say it's between 5.5 and 6 years if memory serves. That seems a little long. I think people could benefit from maybe shortening that up a little bit. Now, I don't know how to do that. You know, I know in some places, I know the standard is 2 for some applications. I know some places it's 1. I wonder if that could take, you know, someone could take a look at that. I don't know if 5.5 to 6 years or even 7 years is necessarily ideal. So I think shortening that would be helpful. I think kind of the advice that I got, people should aim for singles and double, more junior. You know, not everything needs to be a home run.
Okay.
Again, that was very good advice I've received, and I kind of still follow that process. I mean, I don't think I have any senior authorships that are more than 5 figures, maybe. But like, it's not, it's good advice. It forces you to focus your story more.
Yeah.
So it's kind of like one side. I think, you know, transitioning to industry or that kind of thing. I don't know how often people look at industry postdocs as a way to get exposure. A lot of universities now have tech transfer offices. Is it possible to do a postdoc or a PhD and spend some time there to kind of get exposed to what the external world looks like that isn't, you know, writing R01s?
Yeah.
I think that's helpful. I'm sure other things will occur to me. I know one thing that we do at Cabaletta, and this is more for non-terminal degree scientists. So we're in Philly next to Drexel. And I say Drexel because we get a lot of their co-op students. And that's actually worked out very well for us. We've had some folks come in the door, you know, to the co-op until 6 months of work. And then we've then offered them positions. It's worked out really well. I've been very impressed with the students that come out of that. Now, I don't know if every place does it. You know, obviously we're in Philly, so there's that. But
Philly's amazing.
I'm biased. But those are some ways I think, you know, in transitioning to industry or you know, it's important to learn the fundamentals of science and, you know, if you're a PhD level scientist, you know, how to do independent experiments, how to like kind of come up with a research concept and take that forward.
I think that's what you're saying. Like, be streamlined, be thoughtful about the question that you're answering and the studies that you're running to answer that question. So if someone came to you one day saying that they wanted to be a CSO one day, what advice would you give them?
I think so whatever the field is, know your science. It's hard to be a CSO if you don't know your area of expertise. For me, that happens to be T-cell immunology. It's kind of what I know more about than other things. So there's that. I think the other thing is learn what it is. Drug development's a spectrum. Everything from, hey, I've got this idea, hey, let's do a clinical trial, does this clinical trial work, to what does a BLA look like? If you want to be a CSO, figure out that first half. You know, learn how to go from idea to tool compounds to preclinical candidates. Learn what an IND is, what that process is, the submission piece, you know. Not all CSOs are involved in kind of the clinical side, because that's kind of variable. Not everyone has to do that.
So that's actually a question I was going to ask you is, do you think that somebody needs to have the clinical training, the MD, to be a CSO?
No, I don't think that's the case. I mean, I happen to have one, so I realize the weirdness in me saying it like that, but I've met plenty of CSOs who don't. I think, you know, as far as like clinical development, the job is to be a good partner to your CMO and say, hey, I think this might work in someone. I mean, obviously to be on working this in what I just said, but like, that's the general concept.
Yeah. Well, I think there has to be an appreciation of what it takes to get into a patient. Yeah. Like, there has to be an understanding of like, what is necessary to run a clinical trial? Why is it important for us to test the safety, efficacy, so forth? Exactly. And then like, what does this look like actually going into a patient? So like, I do think there's a level of understanding that would be beneficial from like a drug discovery standpoint and a research standpoint, just so that, I mean, that's just my perspective. But.
No, I mean, again, maybe I'm oversimplifying, but again, you have to know what that looks like. Yeah. You have to really know that in depth. That's probably up for discussion, but you have to know this is a candidate, or I know how to get to a candidate that I think could be bioactive in someone and be relatively safe.
Yeah. And when we say candidate, we mean actually like a drug or a
Yes.
molecule or an entity that could actually get into a patient.
Exactly.
That is optimized to get
Yes,
into a
yes.
patient.
Not the first idea. It's like, oh, I can inhibit this kinase,
Exactly.
but then I inhibit 10 other kinases that are needed.
All right, my last question for you.
Okay.
While you have a number of published papers and patents under your belt,
Okay.
if you were going to be known for one thing in this life, what would it be?
Oh, hope I'm a decent dad. But professionally, well, I like to think I'm a decent T-cell immunologist who can develop a drug, get it into a person, and then tell you why it works in a person.
Awesome. Well, thank you so much for this conversation. This was so much fun.
No,
It was such a pleasure to talk to you and to get to be here with you.